All right, so for this one, we're still going to be looking at B2.1, which is membrane transport, but this one's going to be a quick one, describing how different transport strategies are used to move materials around a cell and across a membrane. So we're still focused on this form and function, where we're thinking about how does the structure help in the cell doing its job. And we'll be thinking about pages 214 to 218 here. So diffusion is the first type of transport. It's going from a high to a low concentration of particles. This is going to require no energy. And across a membrane, non-polar molecules are going to pass easily. Something like oxygen, for example, in alveoli of the lungs, the oxygen is going to pass through, carbon dioxide will pass through. These are because these are non-polar molecules. But if you have a polarity of some sort, they're going to pass through at lower rates. So for example, water can pass through a membrane, but if it needs to do so quickly, then it's going to have problems. And so there are special proteins in the membrane called aquaporins that will allow water to travel more quickly. But the key here for diffusion, and I often use the example of someone coming into the classroom wearing a lot of perfume, and then eventually you'll be able to smell them, even if they don't flap their arms like a chicken. The particles are moving from a high concentration, high perfume to low concentration across the room strategy, and it requires no energy. Facilitated diffusion is also high to low concentration. This also requires no energy, but it does require proteins in the membrane. And so ions and polar molecules require protein channels, and that's going to be specific to what's being transported is specific to the protein. In the case of osmosis, osmosis is movement of water. And so osmosis can always happen with water because it's small. But if it needs to move quickly, we need aquaporins. And those are water channels to help the water move more. These water channels prevent hydrogen ions from passing also by using a positive charge at the channel gate. And the way that osmosis works is that if we have more solute in the solution, so that might be like more sugar, more salt in the solution, then the water molecules are affected by those solute molecules, and it's going to make it harder for water to move. So water is going to move from an area of low solute concentration to an area of high solute concentration. So if we had more sugar molecules on one side of the membrane, then through a semi-permeable membrane, the water is going to move from the low sugar area to the high sugar area. And then we have active transport. Active transport is moving actually in the opposite concentration. So from a low concentration to a high. So you can think of that as like that guy on Japan train platforms that push more people into the train. He needs to use energy to do his job. So low concentration to high requires energy and protein pumps. We are stuffing more molecules into a high concentration location. And those particles are only going to move in one direction, though the protein structure can change so that certain molecules move one way and then other molecules move the other way. So that's true of a sodium-potassium pump, which is used in neurons. Sodium moves one direction, potassium moves the other. But in both cases of movement, they're still going to need to use ATP, which is the energy to move those molecules. Exocytosis and endocytosis is HL, and the way that this works is endocytosis and exocytosis are used to carry materials in vesicles in and out of the cell. And so endocytosis is going to be taking in molecules from outside. And so proteins and antibodies, for example, are brought into the baby's body across the placenta, and they come into the membrane here. And proteins in this membrane are gonna pull the membrane in and then pinch it off. So it can make a little vesicle. If you have vesicles going towards exocytosis, then these membranes are gonna join together and open, and the membrane from the vesicle is actually going to join to the plasma membrane, and those materials will be released. This is what happens in the case of neurons firing neurotransmitters to the next neuron. So secretion is the definition, the release of useful materials from a cell. And so exocytosis and endocytosis are being used for secretion or secretory capacity. So to summarize, we have active transport, passive transport via facilitative diffusion, osmosis, and exocytosis and endocytosis, which you should be familiar with all of these. And that is it for this video.